Okay, so today's topic of discussion is second week of development. Previously, we discussed the first week of development in which the zygote that was formed was at two cell stage, and then mitotic divisions occurred here, and then it led to the formation of a four cell stage, then eight cell stage, and finally a morula that was the 16 cell stage. This morula, after accumulation of fluid inside, becomes a blastocele, a blastocyst. Having a blastocele, this cavity is the blastocele cavity, blastocyst cavity, and this structure is the blastocyst. By the end of the first week, that is the seventh day, it is implanted into the endometrium. Then the second week of development starts, and the important stages are the day 8, 9, then 11, 12, and the day 13. What happens on day 8? Day 8 is the time when the blastocyst is just partially embedded in the endometrial stroma. Now, as I've told you before, that there are two layers, the trophoblast and the embryoblast. The trophoblast is differentiated into two layers. A cytotrophoblast, that is, as you can see, this dirty green color cells. This is the cytotrophoblast, and it has a single layer of mononucleated cells. And the other layer that is formed from trophoblast is the syncytiotrophoblast, and this is a multinucleated zone that has no mitotic activity going on, and this cytotrophoblastic layer has mitotic activity going on, and eventually we'll see in later on stages that this will penetrate into this syncytiotrophoblast because of its mitotic divisions. Now, the cells of the inner cell mass, that was the embryoblast, that differentiates into two layers. These two layers, this yellowish layer is the hypoblast, and this bluish layer is the epiblast. The epiblast has high columnar cells, as you can see, and the hypoblast has small cuboidal cells. And there is a cavity that is formed inside the epiblast layer that is called the amniotic cavity. This whitish structure is the amniotic cavity. This is all about day 8. Now on day 9, what happens? That the blastocyst is more deeply embedded into the endometrium, and the penetration defect is closed by a fibrin coagulum. Next, what happens is that small vacuoles appear in the syncytium. This dark green part is the syncytiotrophoblastic, and this dirty green is the cytotrophoblast. So in this syncytium, this dark green area, these vacuoles, empty spaces appear. They increase in size and they join together and form lacunae. And this stage at the ninth day of development is called the lacunar stage because of the development of lacunae. Next, what happens is that this hypoblast, it gives rise to a membrane. This yellowish structure is the hypoblast, which are a single layer of cuboidal cells. It gives rise to a membrane that is called the Husser's membrane or the exocoelomic membrane. And this yellowish membrane is the Husser's membrane, and it lines the inside of the cytotrophoblast. Next is the day 11th and 12th. What happens on the day 11th and 12th? That the blastocyst is completely embedded in the endometrial stroma now. It is not partially embedded, it is completely embedded by now. And the trophoblast, as I've told you in the previous slide, had the lacunae, hence the lacunar stage. That was the day 9. That was the lacunar stage. Now what happens is that the syncytium, it penetrates inside the endometrial uterine vessels, and it erodes the maternal sinusoids, and the maternal blood starts flowing inside these empty spaces, and it leads to a development of uteroplacental circulation. So day 11th and 12th is important in the sense that the uteroplacental circulation develops at this stage. And what is uteroplacental circulation? That the syncytium penetrates inside the maternal sinusoids, maternal blood starts penetrating inside the lacunae, and these lacunae inside the syncytium becomes filled with blood, and it leads to the development of uteroplacental circulation. Another thing that happens in the day 11th and 12th is that the yolk sac, the inner surface of the cytoplasm, this area, it gives rise to an extraembryonic mesoderm. Now, extraembryonic mesoderm is lining a cavity that is the extraembryonic cavity, which is also called the chorionic cavity. What happens is that this extraembryonic mesoderm, this peachish structure is the extraembryonic mesoderm. Spaces start forming inside it, and these spaces join to form a cavity that is the extraembryonic cavity or the chorionic cavity. Now, this extraembryonic mesoderm has two layers. The one that is adjacent to the cytotrophoblast is the extraembryonic somatic mesoderm, and the one that is adjacent to the primitive yolk sac, this is the primitive yolk sac. So the one, this inner lining that is adjacent to the yolk sac is called the extraembryonic splanchnic mesoderm, and the one that is immediately lining the cytotrophoblast is called the extraembryonic somatic mesoderm. Now, finally, we'll discuss the day 13. This is the most important day. What happens in this is that, as I've told you in the previous slide, that the penetration defect is completely healed, but sometimes there is bleeding at the site of the implantation. And as this occurs on the 28th day of the menstrual cycle, it is confused with menstrual bleeding, and it is difficult to identify the expected date of conception because of this. Next, what happens is that, as I've told you in the previous slides, that this cytotrophoblastic layer has mitotic activity going on. So these dirty green layers, this is the cytotrophoblast. It starts dividing and penetrating the syncytiotrophoblast, which is this dark green structure. So this cytotrophoblast, surrounded by a single layer of syncytiotrophoblast, starts penetrating inside, and this structure is called the primary villi. Next is that this yellowish line, this the hypoblast, it produces additional cells that migrate along the inside of the exocoelomic membrane, and it forms the secondary yolk sac or the definitive yolk sac. This is the secondary yolk sac or the definitive yolk sac. This yolk sac is much smaller than the exocoelomic cavity that was formed initially, that was the primary yolk sac. And during this formation, large portions of the exocoelomic cavity are retained as cysts, which are called the exocoelomic cysts. Next, we have discussed this before, that the extraembryonic mesoderm has a cavity that is called the chorionic cavity. Now that cavity has enlarged to this size, and this entire structure is the chorionic cavity, and this small structure is the amniotic cavity, and this is the secondary yolk sac. And the point from where this structure is connected to the trophoblast is the connecting stalk, and this is the point of development of umbilical cord after the formation of the umbilical vessels. That's all for today. Thank you.